Welcome to all participants in this third in a series of webinars produced by Ground Zero Pharmaceuticals Incorporated, a regulatory affairs and product development consulting firm. I am Evan Siegel, the president and CEO of the firm. Today's topic will primarily deal with presenting to investors biotech and small pharma strategies for pitches. The applicability of the presentation will also be for partnering and for other types of interactions between investors, partners, and sponsors of small molecule drugs, biologic drugs, vaccines, and combination products. The idea is, how do you go from early data sets to being able to execute clinical trials in the U.S. or at least under U.S. investigational new drug application? The actual applicability of this presentation will be largely towards any of the countries that are signatories in the International Conference in Harmonization guidelines. So China, India, Canada, Australia, and others will have similar requirements, give or take, for executing early clinical trials stemming from early laboratory-based data. So what is product development all about? We like to look at with a regulatory perspective. No matter how good your data sets may be, your chemistry, manufacturing controls, or product quality, your non-clinical data, or early clinical data, you still don't get anywhere without the regulatory process to drive it forward. So we like to think of regulatory affairs as being the center of the universe, with all the other technical areas being very critical, obviously, to moving a program forward. Well, what do investors do when they come to a sponsor, a university group that has terrific intellectual property, but not a product development paradigm yet? How do those critical data sets and those early concepts and ideas move into a true commercial drug development or biologic development program? Well, the four questions from investors typically are: do you own it? What is the IP and patent status? What kind of marketing exclusivity would you have for your molecule or molecules or your platform? How do you get protection against competitors? The second question is, does it work? What do the preclinical, non-clinical data show from an efficacy point of view, or as we like to say, pharmacodynamic point of view? Do you have in vitro data? Do you have in vivo data in animals? How can you show investors that your product has merit outside of hypothetical constructs, interactions with enzymatic pathways, binding in vitro systems? And what kind of early human clinical data do you have? This presentation is not intended to deal only in the preclinical space, but rather you might have data from approved products. You might have data from other countries coming into the U.S. How do you take those early human clinical data and use those to support your program? The third question is, can you get it approved or licensed? Can you actually get your program through the regulatory process such that the prospects of the program are heightened or enhanced through regulatory certainty or more regulatory certainty? And can you get special status for your program? Orphan drug, fast track, breakthrough designation, rare pediatric disease designation, pediatric exclusivity. What kind of programs can you use to enhance the prospects of the program being considered very critical, perhaps serious or life-threatening illness with an unmet medical need? Those kinds of programs of which you can avail yourself in the U.S. Allow you to perhaps get some of an edge in the regulatory environment. And the fourth question is, can you market it? What kind of competitive arena are you operating in? Can you sell it? Can you get thought leaders, key opinion leaders, others to actually prescribe your product. So that very important question is sometimes forgotten in the euphoria of the concept and the early non-clinical data and even early clinical data. Can you market the product once approved? How does it stack up against competitors? Well, in terms of do you own it, obviously the IP and patent status are critical, but not absolutely necessary in all cases. You might have a product that's out of patent, but because you have such powerful data to support the product and because you get marketing exclusivity, sometimes the marketing exclusivity bars the entrance of another product similar to yours for a very long time. Patent status is critical, but so is marketing exclusivity. And again, orphan drug status in the U.S. confers 7 years of marketing exclusivity for the combination of your product and a specific indication. In Europe, it's a 10-year marketing exclusivity. Pediatric exclusivity can give you 6 additional months of marketing exclusivity. Additional indications, some call those greening of patents, some simply call them extensions or line extensions. Additional indications can give you some additional marketing exclusivity. In the U.S., a small molecule for the first indication approved under a new drug application typically confers 5 years of marketing exclusivity. But a new indication for that same product can confer 3 years, and a new indication after that can confer 3 years. So you can green your product by adding additional indications. Biologics have a 12-year marketing exclusivity initially in the U.S. Very, very powerful. 5 years small molecule, 12 years biologic. And the timing of market entry can be important when you integrate the issues of patent exclusivity or patent status or patent protection versus marketing exclusivity. So sometimes your program can be oriented and designed to make the most use of both patent exclusivity and marketing exclusivity. And then jurisdictional differences actually more complicate than help. There are some jurisdictions which are tougher for biologics, for example, in the U.S., where you do not get 12-year marketing exclusivity for first molecule, first indication. So you have to look at jurisdictional differences when you discuss with partners or you discuss with out-licensed entities or you discuss with investors your program. Does it work? Obviously, the quantity versus quality of data equation is very important. Lab notebooks really give you very little support in the regulatory environment, at least, compared to reviewable reports. So you really need to think about how your data are presented. The greatest investigator, the greatest scientist using students or graduate students in a university laboratory does not necessarily generate commercially usable data without a lot of massaging. So think about looking at lab notebooks and summaries and slide decks, which, by the way, I hate. And slide decks give you a great summary of your information, but when you're looking for detailed data, endpoints, quantity of data, statistical significance, clinical relevance. When one creates a slide deck, one has to think very carefully about how those data are presented and whether there's a lot missing from the typical slide deck. And in vitro versus in vivo non-clinical data, except in the case of certain antimicrobials, antivirals, antiparasiticals. It's generally better to have in vivo animal data than in vitro animal data to support your proof of concept moving forward, but both can be useful. The selection of animal models. What's a relevant species? Do you have the same receptor in the animal model compared to the human? Do you have similar metabolism, similar pharmacokinetics? You have to look at the relevance of animal models in selecting them to support your concept. The number of animals. 5 mice didn't die. Oh my God, we have something approvable, is not really a believable argument. We see this all the time in press releases from small and startup companies. 5 mice didn't die. The product is safe. Well, 5 mice didn't die doesn't mean the product is safe. It's a bare start to supporting your safety and, for that matter, your efficacy. The doses administered and controls used, all pharmacodynamic studies should have controls. As a matter of fact, in general, all scientific studies should be controlled. There are clinical trials which can be open label. They can be uncontrolled if they're in serious life-threatening illness with a well-established pathway for disease and natural history of disease where you can't ethically use a placebo or a control, but there are ways of getting around that. In general, scientific assessment of a new molecule should always have controlled studies involved. And then human data. Do you have anecdotal versus open or controlled data, as we just discussed? The choice of comparators. Do you use standard of care? Do you use standard of care and vehicle placebo? What do you do in terms of establishing your data sets? These are all very important. And the relevancy of endpoints selected, very important. In oncology, the concept of response rate, overall response rate versus progression-free survival versus disease-free survival versus overall survival. The choice of and relevancy of endpoints selected will help determine the strength of your data sets when you're talking with partners or you're talking with investors. The third issue is, can you get it approved? What's the regulatory status? Have you gone through a pre-IND consultation? Do you have an IND open or are you planning it? Do you have an IMPD and CTA, clinical trial authorization, in Europe or Canada? Do you have a CTN, clinical trial notification, in Australia? Have you gone through a regulatory process? That will be very important in gaining additional interest from partners and or investors. What is the magnitude of effect, early versus late studies? Are you doing a dose response study? Are you doing a single escalating dose study or a multiple escalating dose study? What is the statistical significance and clinical relevance of your data so far? Do you have a single versus multiple indications? Obviously, multiple indications, multiple products are very attractive to investors, but you may have only a single entity, a single molecule. Can you green that for multiple indications logically? Is there a common pathway for oncology indication where you aren't stuck with one oncology indication, but you can logically use the product in multiple oncology indications? What kind of special status and designations do you have, as we discussed earlier? And you really need to establish timelines, costs to get to those, to get through those timelines, and key milestones. If you don't have a logical development program from early preclinical or late discovery through phase 1, at least possibly phase 2, you'll get less interest from investors because it really won't tell them you know what you're doing. So to show that you know what you're doing, you should have either internally or through carefully selected consultants the development program with costs, milestones, and timelines applied. The fourth question from investors is, can you market it? What's the competitive arena? Is it indication specific? Is it platform specific? Are there multiple competing platforms where you need to show that your platform has more utility, more possible spin-outs of products and indications? What about key opinion leader and investigator input? If you don't have key opinion leaders on your side, it's pretty hard to compete effectively. If your investigator pool is not that interested in investigating your product, they may agree to do clinical trials, but they're not going to put them at a high priority. So your actual ability to randomize and enroll patients may be limited, which means your timelines expand, which means your maintenance costs expand, which means that your milestones won't be met in the times that you'd like them to be or have promised to investors. It was very, very important to consider those issues. Patient compliance. Is the product difficult to dose? Is it being given a suppository form or intraperitoneally or orally or injected or topically? Those are very important in terms of patient compliance. And then manufacturing, shipping, storage, and delivery. If you have a tailored individual biological therapy, which is made at the patient's bedside or in the hospital pharmacy, that's very different than an off-the-shelf product, which can be consistently made, stored, and shipped as needed from a central depot. So it's very important to consider those aspects when you push your product and your program to partners and investors. There will be region-specific issues such as cost. Medical practice differs in many different regions. And the cost factor is very important. America is the most expensive region in the world for small molecule drugs and biologics. So one has to consider, if you're targeting Europe, you're not going to make as much money per dose typically in Europe as you do in the US. So you have to consider that marketing strategy. Obviously, you have to be humanistic about it, and you want your product to get to patients in need, particularly for serious or life-threatening illness with an unmet medical need. But you also need to consider, from a commercial point of view, the regional issues. The ease of conducting clinical trials in different regions. You may have previous approvals for similar products, which can help or can hinder, because some authorities will say, well, we've already got a couple of good products. Why do we need to have yours in the region? Investigator buy-in, we mentioned earlier, very, very important to get enough interest in your product that you can execute clinical trials effectively, quickly, and efficiently. And what's the risk benefit for the program? You really have to consider risk as well as benefit. Inventors love to talk about the benefits of their programs, and they rarely talk about risks. But every investor worth his or her salt will look at risk benefit, the probability of approval, the probability of being able to market. In the case of formularies, consistent formularies such as one has in Australia in the PBS scheme, if you're not enrolled in that scheme, even if the TGA approves the product, it will be very difficult to sell that product in Australia because the government won't reimburse it. The government doesn't reimburse it. The private insurance scheme is not nearly as developed as it is in the States and some other regions. And so if you don't get on that PBS scheme because your product is not seen as having an appropriate risk-benefit profile or is too expensive, then you won't get much of a hearing in that region. And that's changing to some extent, but you do have to consider that in your marketing strategy. Well, we talked about reviewable reports rather than lab notebooks. What is a reviewable report? Well, the number one lack that we see in dealing with university or clinic or hospital-based programs, however magnificent in concept, however terrific the investigators are, the scientists are, and their track record may be in getting publications or maybe even a Nobel Prize, if your reports are not reviewable, you're not going to get a regulatory hearing of any consequence. So a pharmacology report would be typically involving primary pharmacodynamics, secondary or off-target pharmacodynamics, and the ratio of on and off-target effects. Safety pharmacology should be GLP, good laboratory practices. And so you should use either your own facility, which may be GLP certified, or a good contract research organization that does GLP safety pharmacology. And the same thing with primary toxicology. Nothing substitutes for toxicology and safety pharmacology. 5 rats didn't die with no histopathology won't get you anywhere. So you really need to consider the cost and time and incorporation of these studies into your schema. ADME for most products, absorption, distribution, metabolism, and excretion. Bioanalytical methods development, where your product, whether it be a biologic or a small molecule, can be assessed and analyzed in blood and body fluids, as well as any metabolites stemming therefrom. So these are the types of reviewable reports. Here is an example of a pharmacology report from early laboratory data. You should have a prospectively written protocol with all protocol amendments, or at least a detailed methodology incorporated in the report. A detailed description of study design, including the animal species and models description of the test system, dose levels, detailed procedures for test administration, results for all parameters evaluated for each animal on study. We often don't see this. We hear, oh, go to the lab notebooks. Lab notebooks are not reports that are reviewable by, frankly, either investors or partners or the FDA or other regulatory agencies. A disciplined report is necessary to support a pharmacology investigation. And an analysis and interpretation of the study data. Typically, you'd see that in most entities stemming from these laboratories because they're used for developing publications. But I can't emphasize enough, stop with the publications and start with the reviewable reports, because those reviewable reports are what the regulatory agencies need to see. So you can generate as many papers as you want, but the first thing you should have is not a paper, but rather a pharmacology report from which publications can be derived. I'm not minimizing the importance of peer-reviewed publications, or for that matter, the fact that they can be used to help support a development program, but they do not substitute for these types of pharmacology reports or toxicology reports or safety pharmacology reports. So now moving towards the interactions with regulators. Generally, when you have a new chemical entity, a new biological entity, or you have a new indication for an extant biologic or small molecule entity, it's very wise to take your program to the FDA for a pre-IND meeting. In Europe, for the EMA, they're called scientific advice meetings, very, very similar, conducted differently, but similar. In other regions, you can do this as well. This meeting basically gives your program a hearing before a very key regulatory agency. Investors like to see that you've gone to the regulators, because the regulators are the ones that really tell you what your program is doing for you, what you may need additional before you can enter into human clinical trials, and what kind of deficiencies they see moving forward into the later phases of the program. So it's very key to identify studies that will support the initiation of clinical trials. This de-risks the IND submission. Resolving potential safety issues, very important. If in fact your early data show that there may be a safety signal, that can be resolved with additional studies or possibly a different kind of analysis of the program to date. Discussing available methods to enhance development, as we discussed earlier, orphan drug, fast track, breakthrough, the animal efficacy rule, which briefly is for a product intended to cure, mitigate, or prevent human damage from biological, chemical, or radiological warfare agents. You cannot ethically test the efficacy of your intervention in humans, because you can't expose them deliberately to radiation or to biological warfare or chemical warfare agents. So what you do is you do human safety studies just for safety purposes, and you do efficacy in an animal model, such as a highly irradiated rat or dog, such as a one or more animal species exposed to a small and increasingly large doses of chemical warfare agents. And the same thing with anthrax, for example, biological warfare agents such as anthrax. So what you do is you're able to use a hybrid of animal and human testing to actually support safety and efficacy of your product. A rare pediatric disease designation and regenerative medicine advanced therapy designation. This is for a regenerative medicine such as a stem cell product. You can get this designation, and it helps you get a facilitated hearing with the FDA reviewing division that deals with your therapeutic area. The pre-IND meeting package. You submit a letter, typically, to the FDA, and then once they grant the meeting, and they generally will, you submit a pre-IND meeting package, which contains background information on the drug, the CMC, summaries of available non-clinical data. And you don't have to have a lot for a pre-IND consultation. The more you have, the better you are in getting a very full hearing from the FDA. But you don't have to have 5 tox studies to enter into a pre-IND consultation. And you want to have specific questions to be discussed at the meeting. You don't say to the FDA, what do you think of our program? You say, we have done GMP-like manufacture of our clinical trial material. Our study is intended to be a phase 1 or a phase 1/2 study. We have analytical data. We have a certificate of analysis. We're moving towards GMP for phase 2. Does the division agree? So you basically tell them what you've got, what you're planning on doing, that you feel it's necessary and sufficient to move ahead. Does the FDA agree? And that's how you couch this. I have to say, in the COVID situation ongoing right now, the FDA has abrogated for COVID-related products the idea of a letter requesting a pre-IND meeting and then a package following a few weeks later. Rather, they suggest that you do a pre-screening consultation that takes a few hours, basically, a day or two with the FDA. And they will clear you to submit a pre-IND meeting package and letter in one. We just had one for COVID. We got an initial response from the FDA in 12 hours to a request for consultation. We submitted a little more information, and in about 24 hours, we got a contact in a particular reviewing division at the FDA, and a promise that they would get back to us quickly. They did that in about 72 hours. We submitted the meeting package and letter about a day later, and a week later, we got a full written FDA consultation on that pre-IND package. So it's a very facilitated approach right now for COVID-19 related, either antivirals or immune modulators that could ease some of the respiratory autoimmune or highly reactive effects of COVID-19 disease. So it's a little different, but what I'm discussing here is more the typical approach for an oncology product or a CNS product or an antiviral or an antibacterial or cardiovascular drug or a coagulation drug or the other typical both CBER and CDER therapeutic products. Well, what are modern regulatory strategies and how do they feed into this? You should realistically review your product and indications. If you don't do it, the investor group or partner will do it for you. Look at emerging safety and efficacy signals frequently. Don't get lulled into that feeling that your product is wonderful and it's going to cure all cancer. Meet early and often with regulators. We can't state enough that avoiding the FDA and other regulators is really a bad strategy, because then you're operating in a vacuum. And since the regulators are the ones that help move your program forward or not, you might as well tell the truth and hear the truth back. Consider new clinical approaches, a phase 1 expansion cohort. You don't have to do phase 1A, phase 1B, phase 2A, phase 2B, all this kind of malarkey that's been around for 50 or 60 or 70 years. You can combine phases. You can have multi-phasic studies. As long as you have a logical approach, appropriate stopping rules are identified, the identification of dose-limiting toxicities in clinical trials. It should be scientific, it should be logical, but it can move faster than it has heretofore. All this business about every new drug takes 10 or 12 years to develop is crazy. We've seen drugs developed in 2 or 3 years now because of these new approaches by the regulatory agencies, working smart, having the highest quality data up front, spending more money up front, but getting the best quality data in the fastest time, and meeting often and early with regulators. Multiphasic protocols, adaptive designs, targets and markers across tumor types. This is becoming very important right now. Despite the genesis of tissue or identification of tissue where a particular tumor may have derived, You can now look at common markers, biomarkers, genetic markers across tissues, and the tumor types may be the same. So you can actually target many more indications per product than you used to be able to do. And sometimes you can use a trial that has multiple tumor types, gastrointestinal stromal tumors and certain types of breast cancer often have common markers. So working smart by looking at those common markers in very scientifically based work is one way to go. And that's a modern regulatory strategy that not only has clinical implications, but earlier approval implications and multiple indication implications as well. Fast track designation, simply put, this is for serious illness. It is indication and product specific. You have to demonstrate an unmet medical need. You need preclinical data and increasingly, preferably early clinical promise based on data to get the FDA over the line with a fast track designation. You submit as a formal submission, allow a 60-day review window, and if the designation is not granted, you can reapply, bolstering your present package with additional data. And the FDA will provide specific suggestions. Well, what does Fast Track do for you? It actually gives you a facilitated approach at the FDA, a review team which will probably give you more meetings and more informal hearings than you normally get, given the busyness of the reviewing divisions at the agency. And you have the possibility of a rolling marketing submission at the end of your program, where you can actually have the modules, the CTD modules of your marketing application reviewed in train instead of simultaneously. Breakthrough therapy is a super fast track. The molecule can be alone or in combination with other molecules. It's, again, serious illness. You have to make a comparison with US approved therapies, either approved, cleared, or licensed. That's devices, biologic drugs, or small molecule drugs. You have to have a substantial improvement over existing therapies in one or more clinically significant endpoints. So in this particular case, you have to have clinical data which show improvement over anything you could legally get in the US, or at least approved in the US. And that can be either for safety, efficacy, or both. A lot of people who get excited about breakthrough think you have to show efficacy beyond anything that's approved, cleared, licensed. Actually, it's efficacy or safety. But if you're using safety as the comparator, then your efficacy has to be approximately the same. So it's a fairly stiff marker, a fairly stiff comparison has to be made. You have to use clinically significant endpoints for that indication. Again, the request for designation is a formal submission. There is, for breakthrough, unlike fast track, the option of a pre-request, which is encouraged by the FDA, where you submit a 2-page summary of your program and say, we believe this has merit in breakthrough. Do you agree? The FDA will get back to you typically within a few days and set up a 15 to 30-minute telecon within, give or take, a week or two. And you actually will hear from the FDA what they think about your pre-request submission. If they say, well, we think you need a few more patients to demonstrate this, you should take that to heart. You may submit the formal designation request, but most likely, if you haven't bolstered it, you won't get it approved. So it's good to listen to the FDA advice, a few more patients, a different indication, an additional indication, etc. There's a 60-day calendar review window for fast track, for breakthrough designation request. And if granted, FDA will set up a specific review team, including the division director or his or her second, to actually work with you as the sponsor and move this through. They'll help you physically design clinical trials. They'll meet with you, these days not in person, but in general in person, to help you design the rest of your program to get it over the line. Statistically, breakthrough designated drugs virtually all have priority review They have been improved in as little as 6 weeks instead of the 6 months promised. And typically 3 months is very common. And the proportion of breakthrough therapy drugs approved is much higher than similar non-breakthrough designated drugs in the same therapeutic category. So basically, breakthrough is very, very powerful, and it gets investors crazy. They love it. And partners as well. I mean, you can sell your company or you can sell your product for a lot more money if you're granted breakthrough designation. But it's not easy to get. Well, what do you need for an IND for clinical trial initiation? An IND is necessary in the U.S. if, one, you have a new chemical entity that you wish to test for the first time in humans in the U.S., or you wish FDA oversight of a clinical trial in another jurisdiction, Australia, Europe, etc., so that you get the benefit of FDA advice, and at the same time, you can perhaps execute your program a little bit more cheaply in another region. Or you have an approved product for a new indication that would change the labeling. So you can have an approved product, you want the label to be expanded for a new indication. So if you're going to test in the US or under FDA auspices, you need an IND. The IND structure is simply this. It's the common technical document, which is now universal worldwide as a document that suffices for marketing applications, and in a modified version in some regions, but in the U.S. exactly the way it is laid out as common technical document for an IND. Commercial INDs must all be eCTD. They must be submitted electronically. They must be in CTD format. Investigator INDs, investigator-sponsored trials, do not need eCTDs, but if they're converted eventually to commercial INDs, you need to convert the whole thing to an electronic form. So you might as well start out as an eCTD submission. Module 1 is region-specific administrative information. Module 2 is the summaries of quality, safety, and efficacy. Module 3 is the quality with the full CMC package. Module 4 is non-clinical study reports. Module 5 is clinical study reports. For those of you used to Europe, an IMPD is kind of like a mini IND, and a CTA is an individual clinical trial authorization that contains an IB, a protocol, and the IMPD for each country. The IMPD does not need clinical study reports or non-clinical study reports. The IND does, so that you're building essentially a marketing application dossier from the first IND submissions in the US. So what do you need for chemistry manufacturing controls, or commonly called product quality? The typical phase 1/2 IND package includes documentation of the material, characterization, strength and potency information, stability data sufficient to cover the dosing regimen for the first clinical trial under the IND, the drug substance and drug product data, manufacturing process diagrams, tentative specifications, what's the purity intended for this first clinical trial material, what kind of impurities and contaminants do you have, what's the maximum allowable impurity and contaminant, and what are they? What are the in-process controls in manufacturing? And they can be very briefly stated for this early IND package. And what are the analytical methods do you use? Are you using mass spec, GC mass spec? Are you using HPLC? What are you using to assess these analytical points? Preclinical studies, what do you need? GLP and non-GLP. All primary safety studies intended to support human clinical trials under an IND must be GLP. Pharmacology studies and ADME studies do not need to be GLP. So it's cheaper to do a non-GLP study, although you really should pick a quality provider that is GLP-like, so the FDA doesn't have a lot of questions about the reports stemming therefrom. The toxicology, safety, pharmacology must be GLP compliant, and they provide primary support for clinical trials. There's an exception. Some studies employ a specialized test system which cannot fully comply with GLP. So you simply explain why you can't comply with GLP, and the FDA will typically accept that if scientifically based. You identify the areas of noncompliance and evaluate their significance relative to the overall safety assessment provided by that study or those studies. The typical early IND package includes pharmacodynamic studies, the GLP tox mentioned earlier. Typically in both rodent and non-rodent, the one species rule can enter in when you have serious or life-threatening oncology or other indications. You have promise in your pharmacology data, and so the FDA may waive the two species rule. And for certain biotechnologically derived products where you actually don't have more than one relevant species from a tox point of view that really is predictive at all of potential human risk, then you don't need the two species in those cases. The safety pharmacology panel can be combined with tox studies in some instances. To enhance the potential for success with this preclinical or non-clinical data, you should generate only high-quality data. Forget the large number of exploratory studies. University researchers and professors love to do all sorts of exploratory studies and get lots of papers out of it. This is not a commercial program, and it should not be done. If you have those papers before the program moves over to a commercial sponsor, fine, you can use those to support. But you should not be spending corporate and investor money on aggrandizing university professors, having them generate lots of papers and lots of presentations at meetings, unless they fulfill your need to move your program ahead into and through U.S. human clinical trials under an IND, or clinical trials anywhere under IND. We see this all the time. Grant money, investor money. The investigator does 3 and 4 and 5 little studies, none of which fully support either human potential for human clinical efficacy or human clinical safety. And they keep on generating this work, and the investors in the FDA say, where are the reports? Where are the data that primarily support the potential for human efficacy and human safety? So you need to get away from that university thinking. As important as the universities are in generating terrific intellectual property, the development of a drug or biologic drug or vaccine product should be left to the drug development experts. And consider testing only in relevant species. The seeding studies is similar to what I mentioned a minute ago. Avoid a lot of little studies that don't really mean anything. You need to spend the money up front to do it right. Then you, one, won't have to repeat those studies, and two, you wind up spending a hell of a lot less money 6 months or a year or 2 years from now if you do it right and do it right the first time. And for serious or life-threatening disease, you have facilitated and accelerated approaches, as was mentioned earlier. Even clinical design can often be much more aggressive and get you farther faster when you have serious or life-threatening illness. Well, how do regulators and investors review clinical trial protocols and strategies? Well, the format should be reasonable and readable. ICH-type guidelines for protocols and for study reports are kind of important and help reviewers review the way they like to review by seeing data elements in a certain form, a certain format, and at a certain level of detail. Include the procedural information. How did you conduct, or how are you planning on conducting the clinical trial? What's the statistical analysis plan, at least descriptively? The informed consent, what does that look like? The informed consent should be congruent with the investigator brochure, should be congruent with the IND summaries, should be congruent with the protocol. If they differ, the regulators will say, wait a minute, why are these different? It's the same information, the same approach. Include Data Safety Monitoring Board information as applicable. You may not have a DSMB early, but you may if you're in a very aggressive posture, like for COVID trials right now. They're pretty much all with DSMBs, even in phase 1. And you don't need exhaustive references. Your elements in your protocol should stand on their own. The concordance of protocol with other information, we mentioned that earlier. Safety support is critical. The pharmacology data should support the proposed study. Makes no sense to propose a study that's not supported by pharmacology, pharmacodynamic data. And the manufacturing data should allow for a thorough review of the drug substance and drug product when in comparison with the elements included in the protocol. How are you going to dose the patient? How often are you going to dose the patient? How is the formulation consonant with infusion versus bolus injection? These things are very important, and they show an interface between product quality and clinical trials. Even though most people don't connect them, they are connected. The non-clinical, the clinical, the regulatory, and the chemistry manufacturing controls all interpenetrate and support each other. Well, there are some special issues in the remaining time we have for biologic and biotechnological programs. And I should mention right now that on the bottom of your screen, you have a dialog box. You can ask any questions you want. We'll have ample time for Q&A after the formal presentation. So ask questions as many as you want. Lauren Mages will, upon request, send everyone on this call copies of the full presentation, so you don't have to worry about taking extensive notes, and I am moving fairly rapidly anyway. So special issues for biological biotechnological products. CBER, Center for Biologics Evaluation and Research at the FDA, regulates all cellular products, human, bacterial, animal cells, the gene therapy products, vaccines, antitoxin, blood and blood components in products, plasma-derived products. Those are all regulated in CBER. If you're wondering what about therapeutic biologics like peptide, monoclonal antibodies, those are reviewed in CDER, Center for Drug Evaluation Research. They used to be regulated at CBER. In around 2003 to 2006, the therapeutic biologic products went over to the CDER, except for the cellular, gene therapy, animal cell, live cell, antitoxins, blood and blood products stayed in CBER. Some biological biotechnology investigational products warrant additional containment considerations: pathogenic microorganisms, transgenic animals and plants. They require special concern, and they are dealt with very effectively by CBER in conjunction with you, the sponsor. And those CMC plans should be discussed very thoroughly with the FDA at the pre-IND meeting. Typically in biologics programs, chemistry manufacturing control / product quality issues rise to a level of greater importance very early in the program than they typically do at CDER, which deals with either the straightforward monoclonals, the peptides, or the small molecules which are fully synthesized. And the reason for this is because it's difficult to predict the impact of changes in quality attributes on safety. So form and function from an efficacy and a safety point of view are very much interconnected. It's especially true for phase 1 studies because the comprehensive product characterization you see later in the development program for a complex biologic is not often seen in the early stages. So one has to be very careful to have a good certificate of analysis of your biotechnologically derived or complex biological product such that you have some reason to have confidence that you can reproduce the results you see both for safety and efficacy in early human clinical trials. So that reproducibility with control and documentation of the production process are important. And process development should be fully documented. This is how did you get from here to there? How did you establish your master cell bank? How did you establish your working cell bank? You don't have to deal with these with synthesized peptides or small molecules, but you do with biotechnologically derived complex biologics. With that, I'm going to turn this over to Q&A and look forward to your questions. So it'll be on in your Zoom room, your Zoom group chat box. Now we can cover anything if you want to talk CMC. There we go. Can you explain extending market exclusivity? Good question. Yeah, I mean, marketing exclusivity itself is granted and doesn't get extended anymore in and of itself. However, if you have marketing exclusivity for your first indication, first molecule, whether it be a biologic drug or a small molecule drug, you can extend exclusivity for your product by developing a new indication. In the case of an NDA, for example, a new indication would typically be studied in clinical trials or trials, and then you'd apply for a supplemental new drug application approval. That first supplemental NDA, new indication, same molecule, would give you 3 more years of marketing exclusivity. So you could add the 5 years of marketing exclusivity that you were granted first time around with 3 years for each new indication. Now, what that marketing exclusivity does, because we didn't describe it earlier, is it blocks in the small molecule arena. It blocks another sponsor from submitting an abbreviated new drug application and doing a Me Too product against your product. Once your 5-year marketing exclusivity is gone, that's it. Now, you may still have patent protection, which could block other entries into the market, but the marketing exclusivity is probably the most powerful. Oh, okay. Can you discuss QIDP, a qualified infectious disease product? Yeah, this is a designation which is provided anytime during your IND program where you supply information on your product and you actually have a specific indication for serious or life-threatening infectious disease with an unmet medical need, and your product, or actually your combination product, say a drug device, can be granted QIDP, and it gives you some special handling at the FDA. Can you talk a little more about what investors want to see in a pitch deck about pre-IND IND submission? Which points should be highlighted up front? Well, that's a good question. I didn't really intend to be so negative about pitch deck, but I've seen so many pitch decks that really say very little. They talk about the wonders of the theory and the mechanism of action. Mechanism of action is not unimportant in dealing with regulators and your program. However, in fact, in this country, it is really secondary to having 2 key genre of information. One is safety or predicted safety, and 2 is efficacy. Your mechanism can be absolutely wonderful, be heavily patent protected, but if you don't have safety and efficacy data, then you really go nowhere. So what I say is, do you own it? Yes. Does it work? Okay, there are your data. Can you get it approved? There's your regulatory, and can you market it? There's your competitive environment with all those characteristics that are critical for it. So, with the pre-IND, I would say we have had a successful pre-IND meeting with the FDA. We need very little additional data. The IND will be submitted in 3 months, or We had a successful pre-IND consultation. We tweaked the profile. We added a little non-clinical data, and we submitted the IND 3 months later, and the IND is open. That's more important than, oh my God, we've got a wonderful concept. I'm sorry to the inventors in the audience, but for investors, they want to know, when is this thing going to get on the market? When is it going to build value in the stock market? How is the partnering going? What data and what regulatory support do you have for the program? So that's my comment on that. Let's see, we've got a lot of questions, so I'll try and go in order here. Okay, real-world data or real-world evidence support an IND or NDA. Yeah, the FDA has a major move on, partially because of the CARES Act and other legislation, which has forced us to open up the process a bit more to real-world data, real-world evidence. I guess one good way to put it is if you have anecdotal data from a cohort of patients which makes sense when combined, or explicate the natural history of the disease better than has been done previously, or adding a body of information to the controlled clinical data body of data that you have that is extant, then real-world evidence should be considered even for approvals, because what happens in the real clinical situation in patient treatment situations is sometimes as important and can be even more important than the safety and efficacy profile you see in a controlled clinical trial. So yes, you can support an IND. As a matter of fact, we were doing it for a client probably 18 years ago, something like 18 years ago. There were some real-world data from a group of anecdotal patients that was reasonably well codified in case report forms. And we put those data together, and the FDA accepted those data more like a quasi-clinical trial instead of just anecdotal data. It was serious or life-threatening illness. They don't stand on ceremony when it comes to that. So that may be some help to you, and we can further explicate if you want. Let's see. It doesn't take 10 to 12 years to develop new drugs anymore. Can you expand on this? Well, I'm not saying it doesn't, but it doesn't have to. Because of the programs that the FDA put in place starting in 1987 and then accelerated very heavily in FDAMA in 1992, where industry fees for marketing applications have increasingly supported not only the review of marketing applications, but actually IND programs as well. The FDA gets at least 40% now of all of its funding from fees for establishment, registration, licensing, for marketing application fees, and from approved drug fees every year. And the rest comes from the Department of Agriculture budget. Amazingly enough, it's not the HHS budget, it's the Department of Agriculture budget for the FDA, and that's due to historical reasons starting in 1906. But essentially, with serious or life-threatening illness now, if you do it smart, and if you have a really strong signal of efficacy in animal data, you can accelerate the entire program. Remember I said earlier, phase 1A, phase 1B, phase 2A, phase 1B, phase 3A, phase 3B, phase 3C. All of this is really nonsense for a lot of programs. As a matter of fact, at GCP, we prefer to use the term exploratory and confirmatory. That's it. That's the two types of studies. You're doing exploratory work in phase 1, phase 1/2, and they do confirmatory in what used to be called phase 2B and or phase 3. You don't even need phase 3 for a lot of programs now. You can get an approval with 3 clinical trials perhaps if you do it smart, and if you're lucky, and if your product has a very strong efficacy profile and an acceptable safety profile. It doesn't have to be the safest drug in the world, but if it's highly efficacious and there's an unmet medical need, a lot of the data you used to need to submit, the staged tox data, that goes throughout the program, the heavy-duty reproductive data, the carcinogenicity data. A lot of that is waived for serious life-threatening illness and unmet medical need. So too, the clinical trials process has been accelerated to do it smarter. And so that's why you can compress that, quote, 10 to 12 years. Some people say 15. I mean, look, pharma loves to say, oh my God, it costs $2.5 billion and 13 years. That's crap. If you take a look at the history of drugs in serious or life-threatening illness over the last 5 years, even 10 years, and moving forward now, you look at how many breakthrough therapy designations have been granted in the last, say, 5 years, and take a look at how many of those have been approved in the last 5 years. Clearly, the entire length of the development program is nowhere near 10 to 12 years. So it depends on what you're talking about. And examples are, well, ibrutinib, for example, for chronic lymphocytic leukemia, the CAR-T therapies for AML and other indications. These did not take 10 to 12 years. They didn't even exist 10 to 12 years ago. So there are plenty of examples, particularly I can point out in oncology, but to some extent in rare genetic disease, enzyme deficiency, congenital genetic disease deficiency syndromes. The development programs have been much shorter. So those are some examples. Let's see. The Interact meeting. Well, the Interact meeting has been added. It used to be called the pre-IND meeting, which we used the term 20 years ago, and it was adopted both at CDER and CBER. It was actually physically called pre-IND at CBER until a couple of years ago, and what they decided to do was make it an Interact meeting. And the reason for it was the pre-IND meetings sometimes were really pre-IND meetings. Companies didn't want to blow their pre-IND meeting early in the program, so they would have very early data, and they'd have a pre-IND meeting, and then they could preserve their to be granted pre-IND meeting for later. So what happened was the Interact meeting was sharpened and focused to deal with complex manufacturing issues for a product that needed solving between the regulator and the sponsor before the sponsor could really move forward, or a complex tox issue where you may not have had an apparent relevant species. How do we get around this? Do we need to use a recombinant mouse or a severe combined immunodeficiency mouse model to examine tox? It's not intended to look at CMC, nonclinical, and clinical before you have much data. It's intended for a very targeted discussion with the regulatory authorities at CBER prior to moving forward into the generation of additional data, the planning of your full non-clinical program, and thence your pre-IND meeting. Can you also emphasize FDA and investor strategies for drug delivery systems? Yeah, I mean, drug delivery systems are very important. As a matter of fact, they're probably increasingly important. Depending on how your system is integrated with your biologic or your drug, a drug/delivery system can either be a combination product or it can be a unit dose drug product. So for example, if the primary mode of action of your product is pharmacological and you happen to have the active pharmaceutical ingredient or formulation incorporated in something like a Band-Aid or a bandage, that product generally will be regulated as a drug with a unit dose delivery system. If you have an inhaler into which is inserted a capsule of drug product, that's typically a drug device combination or a biologic device combination product. So the strategy has to be, one, if your delivery system is separate and distinct from your unit drug product, is it cleared in the US under medical device? Is it approved in the U.S. under a PMA? So is it a 510K device, or is it a PMA device? Is it approved elsewhere? Does it have a CE mark, which frankly FDA couldn't care less about? But it means you have a dossier, so you can update that dossier for the FDA. So the strategy of moving your delivery system ahead along with your drug is important to discuss with the FDA. That's very, very important. And sometimes you'll have a meeting with CDER or CBER and CDRH at the same time, like a pre-IND meeting. Or you can have a Q meeting with CDRH to discuss the status of the device and then move that information into the CDER reviewing division or the CBER reviewing division or office. So I can expound on that if you need more than that. Let's see. I think that those are the questions so far. Pricing and reimbursement information. Well, yeah, I mean, investors are highly concerned about pricing and reimbursement, obviously from the point of view of profitability of a company. And let me say this first. Most small firms will not exist as small firms in terms of the product development or the product that we're talking about, our products or platforms we're talking about. Most small firms eventually sell their company or sell or out-license their products or platforms. So pricing and reimbursement for the company itself may be less important, but it's always important for the investors and for the partners because, in fact, investors are always looking for X multiple of their investment dollar in Y years. We've often seen 3 to 5 years, the return on investment in 2 to 4 years. That's been typical in the biotechnology and small pharma area in our experience. Others may have a different experience. The idea of pricing, if your product is cheap to make, but it would command a very high price in the market, that means a high profitability index for the firm and or the product moving forward, even if that product is sold, because that positive assessment carries forward to the next round of investment or investors. If the primary investors cash out, they're going to get a certain ROI, and the next investor group will be interested, or next tier of investment will be interested in potential profitability. So cheap price, cheap price to make, cost of goods and marketing versus high price in the market. I mean, that's the ideal for the strict return on investment calculation, but you won't always get that. I mean, obviously, if a product is really cheap to make, there'll be price pressure on the company not to sell it at a really high price. If the program has been very facilitated and relatively inexpensive, there'll be very little justification in the real world to have a huge high price. You can still get it in some instances if it's the only product in the market or it's the best product on the market, but all of those equations are continuously being made as the investment tiers move on. Reimbursement's very critical, obviously. If a product's not reimbursed, you got a problem. Australia has a tight reimbursement gating. Europe, in the UK particularly, has a very tight reimbursement gating. If your product is marginally better than another product in the market and much more expensive, you probably won't get reimbursement in some cases. And so you have to be careful. In the US, if the product is approved and it's a, quote, good product, end quote, from a risk-benefit point of view, it will typically get reimbursed. I say that not being a reimbursement expert, and you have to take into consideration very heavily Medicare and Medicaid, as well as private insurance, which is polyglot all over the place, as well as other types of programs. So you have to be very careful to keep your finger on the pulse of reimbursement and pricing as your product development program moves ahead. Let's see. I don't see anything else. We'd be glad to wait a minute. Someone else has any more, has another question or two. We've got the time, so if you have other questions, great. As you're thinking, it's important to remember that you need to integrate all these strategies to be successful. That's why the kind of 4 primary questions at the beginning of this presentation, both for investors, partners, expansion of the firm yourself through organic growth, all of these factors need to be taken into account in an overall integrated drug biologic vaccine development program. Okay, here's a question on, if the patent runs out while the product is in the market, do companies then rely on FDA exclusivity? Is this a typical case? Well, yeah, in the old paradigm of very, very long development programs, of course, patent life has been extended in some cases, and of course, the de novo patent in this field now is typically 20 years. So that's a pretty long time. And if in fact the development cycle is decreasing in time, particularly serious life-threatening illness, unmet medical need indications, your patent life will still be there. And at the same time, you can have marketing exclusivity. Yeah, I mean, there are companies that have relied on marketing exclusivity as their patents were running out. You might have a patent with 3 years left on it at the time of approval, and if marketing exclusivity is 5 years, then you get 5 years. In a biologic now, all the biologics regulations for patent exclusivity, of course, quite new. And biosimilars did not officially exist in the United States until a few years ago. And now you have a whole system of development of biosimilars, and there's therapeutically substitutable versus alternative products, but you still have that 12-year marketing exclusivity for the molecule first indication. So it's now polyglot. But yes, companies are relying, particularly in the biologics area now, they're relying on marketing exclusivity for that first approval to really carry them a long way to block companies coming in. Remember that marketing exclusivity is not 100%. There are ways of breaking it, but it's not easy. I don't know if anyone wants to talk about COVID, but I will tell you that if anyone is interested in COVID, it's moving like lightning. It's moving like lightning in the vaccine area. It's moving like lightning in the direct-acting antiviral area. It's moving like lightning in the repurposing of approved antivirals and other drug products as antivirals, and also the cytokine storm and other disturbing immunological adverse effects secondary to COVID-19 moderate to severe disease. That area is moving literally like lightning. The FDA has released something like 5 guidances already on COVID-19 related IRB reviews of changed protocols, remote monitoring, changed ongoing protocols due to COVID-19. That would be for other products, and the COVID-19 effect on clinical trial sites and execution of clinical trials in other areas, as well as facilitation of COVID-related programs directly. Pointers on how to determine if our molecule has clinical value. Yeah, I'll be very straightforward on that one. I kind of do it every day. You can look at precedents in your therapeutic area for approvals, but you can't rely on them in both directions. One, if a product was approved on, for example, overall response rate 10 years ago, it is highly likely that that endpoint will have changed in that specific area to possibly progression-free survival, disease-free survival, time to progression, composite endpoints, overall survival, or combinations thereof. So too, it can go the other way. What at one time required overall survival in certain oncology indications has moved towards progression-free survival or even pathologic complete response or partial response as indicative of that original hard endpoint. So it can be liberalized or it can be tightened up depending on actual experience in the marketplace and in the clinic. So clinical value will be measured by the degree of response, the proportion of responders, the specific clinical endpoints or surrogate endpoints chosen, the reproducibility of the data, the magnitude of effect. So this is a classic one. Let's just talk about overall survival for rare genetic disease, such as key enzyme deficiencies, genetic syndromes, or oncology. If your product against historical control shows one more month of overall survival, that's not generally considered a significant magnitude of clinical effect. If you're in adenocarcinoma, the neck of the pancreas, and you have 12 months overall survival in a clinical trial, where the historical survival is give or take 7 months from diagnosis, you've got something that's considered clinically relevant. And if it's statistically significant, you could possibly get an approval on one trial, or at least an accelerated approval. So clinical value should be looked at from the point of view of magnitude of effect, endpoints, key opinion leaders, because the FDA uses key opinion leaders in advisory committee meetings. They get advice particularly for new chemical or biological entities, or first entity in for a specific indication, nothing had been approved before or licensed. They'll look to key opinion leaders. So you need to look to key opinion leaders in your field and at least use that as part of your assessment of the quote clinical value end quote of your product or potential product. Because those key opinion leaders are the ones who will be prescribing a prescription drug. So you'll want to look very carefully at not only your excitement with your receptor or your in vitro data or even your animal data, but what's the real clinical value of your product? Who are the key opinion leaders typically? How does one approach them? Well, these are the people who, well, for one thing, these are the people who execute a lot of the clinical trials in a particular area. So let's say you're in an oncology indication. All you have to do is look on clinicaltrials.gov for that specific indication at any phase. Even phase 1 is fine. And you'll find who the investigators are and where the clinical sites are. So those are going to be the key opinion leaders, the ones that do a lot of clinical trials. If you're not doing a lot of clinical trials, you can still be an opinion leader. But in general, you're not necessarily seen as one who has a great deal of knowledge, modern knowledge in the area, because you're not keeping up on it. You may read like crazy, and you may actually be a good opinion leader. But generally, the ones that are doing clinical research are generally considered key opinion leaders in an experimental setting. And you locate and approach them by, you would generally use either a consulting group or a CRO that is expert in your field, that is in your therapeutic area or areas of concentration and concern, and you would go to you would go to these people and say, hey, we need 6 investigators for our phase 1/2 clinical in pancreatic adenocarcinoma, and they will find them for you. You could also seek them directly from looking at clinicaltrials.gov, but that's not a good way to do it. Usually you go to someone who's a finder or a group that's a finder that really kind of knows. The other thing is you can look at publications in a particular area. And despite my comment on publications, which is not really intended to be basically negative, people who publish a lot on a specific clinical area are probably considered key opinion leaders in general. So I hope that helps. How early should we involve KOLs? Well, it is generally recognized today that the earlier you at least run your program by a KOL, a trusted KOL, someone who's not going to reveal your information to competitors and others, the earlier you approach that KOL, once you have probably early data. Now that could be early clinical data, or it actually could be good, solid pharmacology, pharmacodynamic data combined with some tox and safety pharmacology data. So you've started to profile your product. You say, well, wait a minute, in a good model of disease, this stuff was gangbusters. You actually can compare your nascent product with other products in the market. There's nothing that prevents you from a patent or an exclusivity point of view from using an approved product in a clinical trial or in a non-clinical trial or an in vitro trial to measure against the biological activity of your molecule. And once you have some early data, it's probably a good idea to involve KOLs and say, look, here's our product profile. That's another thing, doing a target product profile. Here's your market. Here's your ideal data set. Here's what you have so far. Here's what the molecule looks like. Here is your proposed route of administration. That product profile you can bring to an opinion leader and see what he or she says. I mean, it's cheap to get that advice. You know, cost you a few $1,000s maybe, and they'll review your, use the term prospectus advisedly here, but your prospectus for your product profile, and give you some opinion on what they think would be very helpful to have, and including the development program. What should the development program look like? Any more questions, technical questions, questions on investors? Choose your investors wisely. That's something that I would say up front. That you know, be careful how much they want to take, how much they're willing to give, how the valuation of your product is going to go up as your data package improves, increases, and expands. So don't either overvalue yourself, which often happens in small firms that are at the very beginning. I mean, don't overvalue yourself, but also don't undervalue yourself because you're desperate for funding. So it's a balance, and a good investor group will work with you on valuation. Or an independent valuation group can be helpful as well. Another minute or two for more questions, and if not, then we'll close. I don't think there are any more at this point. You know, I think we've answered all the questions that have come in so far. One more minute, and then we'll. No? Okay. Lauren has sent this. Please email her for any future topics. The topic today was actually designed to fulfill several requests we had from the last webinar or two. So we really take your comments and thoughts into account, and we'd be glad to do one. We're actually close to having one finished on gene and cell therapies. So, oh yes, we have one more question. Sure. Feel free to ask it. Have you seen a good university out-license program? Well, there are a lot of, quote, good programs. It depends on what you mean by good. The university out-license programs almost universally do whatever they possibly can to maintain university control, university legal control, and the maximum benefit for the university. So from the point of view of protecting the university, there are a lot of good out-license programs. There are a lot of out-license programs where good technologies are offered for development outside the university, because really, truly, universities are not the place for commercial drug, biologic, vaccine development in general. The University of Queensland has quite a significant out-license program. That's in Australia. The University of Sydney has one as well. The Coling Research Institute there. Monash University in Melbourne. University of Pennsylvania has a significant out-license program. I think UCLA does as well. Possibly University of Chicago. Harvard has had one for a long time, but I don't know exactly how active it is at the present time. So I think if you're looking for compounds, again, it's best to go to someone who kind of knows the lay of the land with that, or approach university technology transfer office, where you know that deals have been done. And the university I mentioned, there have been a lot of deals done. How can we determine a clinical value for a molecule in preclinical phase at a university? Okay, this is where half of this presentation was oriented. And to condense it down, the preclinical phase has many meanings. A pharmacodynamic study in vitro or in vivo coming from a university should have a report, not just a paper or two or three, and not just lab notebooks. You're not going to get the entire story until you see all of the data. Now, sure, you can reconstruct those data from lab notebooks, but it's a hell of a lot more difficult to review them. And we've done due diligence for buyer, seller, or both as an independent third party. And we do not like to see lab notebooks. Sometimes we're forced to. We've actually created reports from lab notebooks when we're paid to do so. But in reality, we like to see reports because it says to us that the university lab knew what it was doing. Even if it's a good data set, a raw data set, a protocol, a results discussion, conclusions paper, you can put that together and you kind of see it or make it as a report. But just having lab notebooks with half a year of raw data in it, to me, that's got very little value, even if inherent in those data, there is value. So you want to see reports. Most universities don't have GLP tox facilities, but sometimes they do what might be called dose ranging tox, and it's pretty good. If those data include gross and histopathology, then you're cooking there, because it means they kind of know what they're doing in terms of pre-commercial data sets. So the clinical value is going to be most determined by pharmacology data in a specific model, a good animal model of disease. That's where you're going to be able to determine clinical value, and preferably with controls and also positive controls. If you see those kinds of data, even if they're not in formal report but can be pieced together, then the clinical value of the assay is probably promising. But just a receptor binding, or, oh my God, the avidity receptor binding is fabulous. That doesn't tell anything to me. I'm sorry. It tells nothing to me. An antiviral in a good in vitro model, sure. An antibacterial in a good in vitro model, sure, that's a good step. But it's not definitive. The animal model of disease pharmacodynamic data set is the first thing I would look at, all of the things being equal. Okay, I think that's probably all the questions now. Thank you all for attending and for your great questions. We look forward to more. We're actually got a presentation almost done, almost ready for webinar on cellular and gene therapy programs, early therapy and gene cellular programs, and how to move those ahead. What are the issues in those fields? And also we'll include some comments on COVID and vaccines included in that presentation. Well, thank you all. Be safe. Have a good rest of the day or evening.